Good afternoon, everyone. We'll now move on to our next forenoon session. It's a moment of extreme pleasure to welcome our honorable speaker, Dr. Reena Pandit, Institute of Mechanical Technology, Mumbai, for our today's session. Ma'am is currently working as an associate professor at Department of Biotechnology, Institute of Chemical Technology, Center for Energy Biosciences, Mumbai. She has contributed a major role in the area of development of R&D laboratory for algal biotechnology, third generation biofuels and biochemicals. She has been rewarded with Bioenergy Awards for Cutting Edge Research BACR Fellowship by Department of Biotechnology and IUSTF. Ma'am is involved in some of the major projects which include growth engineering, media engineering, wastewater treatment, carbon dioxide sequestration, bioreactor designing, biorefineries, and also improving sustainability of algal based technologies. Ma'am has published many research papers and has few patents in her credit. With great delight, I now welcome Mrs. Dr. Reena Pandit to deliver a lecture on microalgae as sustainable energy and its cultivation. I'm sure you all will feel enriched with knowledge after completion of this seminar. Over to you, ma'am.
Thank you. Thank you so much, Navina, for this introduction. And a very good afternoon to all of you. So I assume that you had inauguration section and then there was one lecture which was there, technical lecture which has happened. So we are resuming after break. Uh, so I will start presenting, but my request is that any of you can stop me at any point whenever you feel that you want to ask any question. Navina, are you there?
Yes, ma'am. Will inform you,
Navina,
ma'am,
just
if
a
any question.
just a request to you that if somebody is raising his or her hand, please stop me because since I will be presenting my slides, so I may not be able to have a look at the chat as well as if someone has raised his or her hand. So you can stop me and then I will answer. Read the question for me, I will answer. Okay,
Yes,
because
ma'am,
that I
sure.
would like to have the interactive session. So that's the reason I'm requesting you to just help me out with this. Yeah.
Yes, ma'am.
Yeah. So at the onset, I would like to thank Premlata Madam for giving me the opportunity to be the technical person for the this particular faculty development program. And what I would do, I will start presenting my slides. Let me share my slide. Thank you. Yeah, so I'm Reena Pandit and I am working as an associate professor at Institute of Chemical Technology. So I will just quickly give you the brief about the institute to which I belong to and the department history. So this is my institute. This is Institute of Chemical Technology. It is in Mumbai, Matunga. It is in the central part of Mumbai, and it has history of almost more than 70 years now. We have been there, and we have been catering into and now very recently in past 2 years we have our footprint in Jalna, that is Marathwada part of Maharashtra, and we have also one campus in Bhubaneswar, Odisha. Now, all these campuses and the institute is offering you, the technology courses. So we have 11 technology departments, which includes chemical engineering, food, pharma, oils, and then textile and so on. So 11 technology departments, and we have two center of excellence. One of the center where to which I am work associated with for past more than a decade now, and we have more than 1700 students getting graduated or post graduated every years. Almost we have ratio of 50 PhD students per year. That's the highest PhD student to faculty ratio we have, and we are being funded by state government, and also in addition to that we have lot of private, because we work in very close association with industries, and that's why we have lot of good amount of funding also coming from different industries. Um, So this is the center. So that was the institute. This is the center, which is, you know, present in Mumbai campus, and this is DBT ICT Center for Energy Science, Energy Biosciences. I'll just brief you about the center because the talk has been on the bioenergy, the earlier talk, and you have a few talks based on bioenergy. So I would like to tell you about what all initiatives Government of India has taken in this bioenergy and biofuel space. So this particular center is being funded by DBT, that is Department of Biotechnology, Government of India, Ministry of Science and Technology. So this is almost the center is being funded in two phases. So it was more than 12 years of funding now, and the I think the government has spent, the Department of Biotechnology has spent more than 40 crore in establishing this particular center. Now this is India's first bioenergy center. Followed this center, you have one center with ICGB in New Delhi. Then there is one in association with IOCL, that is Indian Oil Corporation. So that is in Delhi. Then there is one more center, which is, that's a virtual center again, which is wherein you have 7 different IITs coming together. So Pan IIT Center, and the latest is being funded with TERI, New Delhi again. So these are the 5, 6 different centers which are being funded to work in the area of bioenergy and biofuels. So that's the funding which DBT is sponsoring. And in addition to that, Department of Science and Technology, that is DST, has also funded a few centers and few departments for working in this area. So this particular center is established in 2009, and we have no almost more than 50 different scientists which are working in the area of biology, chemistry, chemical engineering. Okay, why I'm stressing upon that the very fact that we have the amalgamation of different people coming from different expertise, because that's what is required. It's a multidisciplinary center and multitasking center. The very reason for highlighting this, that the biofuel space is very crowded and very, very challenging, I would say. Super challenging. And that's the reason why you have to start working on multiple aspects simultaneously. It wouldn't happen that you can do this, okay, I will make this first and this first. No, that doesn't happen. You have to start working parallelly on many different fronts, and with the help of many different kinds of expertise. It's not the number, but it's the kind of expertise which is coming together to achieve the task. That's something is And that's a very difficult model to establish, because we Indians are so used to confining ourselves into a particular area, and getting and not diverting ourselves into, and not focusing on other areas, and not working sometimes, rather many times, not working with people with other different expertise. So that is something would stall our progress. And as the earlier speakers have discussed, that we need to be self-sufficient. The country needs to be self-sufficient in terms of bioenergy, biofuel. And to achieve that, it is very important to do this kind of a multitasking and multidisciplinary approach. That is something, the model which we have developed at the center. Okay. So the objective which was given to us by the Department of Biotechnology was development, demonstration and transfer of cost-effective sustainable lignocellulosic ethanol technology. This was the one mandate which was given at the start, but then the mandate was also extended to building capacity in the field of advanced biofuels and biochemicals. It's that the funding doesn't stop, or rather the mandate doesn't stop just at bioethanol, but it was extended to advanced, because the world is moving so fast in this area that it has to be extended for advancement as well. Bioethanol is something which is our immediate requirement. But very soon we will have, I will discuss that why we would have to, why we need to look at other biofuels, advanced biofuels and biochemicals. So that was the, and it was something which many labs are working. I'm not saying ours is the only center. Many labs are working. But what happens is that whenever we are working, we work at the laboratory scale. But what is more important, and whatever you are able to achieve at laboratory scale, you should be able to take it to the preparative and pilot and demonstrative scale, because there are huge challenges over there. Something which is you are able to achieve in a shake flask, say even at 1 liter reactor level, say you are processing 1 kg biomass, it is doable, it is feasible, but the cost economics, the sustainability and the footprint and all those things would start creeping in the moment you try to scale up, you know, to scale up the technology. Something which works, those controls which are working very fine when you are working at, at laboratory scale may not work the same way when you are scaling them up. So India has to have a translation facility wherein the laboratory scale technologies can be matured and can be taken to the demonstration and commercial scale. That is, there is a huge gap in between, and somebody has to fill that gap. Of course, the funding agencies are looking into those aspects, because that is something more crucial for now, and that requires different set of expertise, and that is something India has to do for the in for a kind of a capacity building in that area. Why we have to do this? Why not? Because see, the our typical trend is that if something is working in US, we can always get buy the technology, license the technology, and can do it in India also. That's not a problem. That's what people have been doing for all these years for many different technologies. Then why we have to think differently when it comes to the biofuels? The very reason is, before that, I'll just take you to the, so these are the state of the art capacity facilities we have at the center, and we are into designing. So we design molecules, microorganisms, we design full scale plants, processes. When I'm saying we, that means the team. I'm not the expert. I am a biotechnologist, so I restrict myself with microorganisms, algae, and molecules. But yes, we have the capacity and the competent people at the center who can, design different processes and full scale plants. And that's the reason I said that if I'm doing something with microorganism to translate it to the full scale plant, I need a different kind of expertise which should be available in house. And this is the 2G ethanol plant which is being demonstrated at Kashipur. So that's the technology which was developed at the center. So this is at Kashipur in Uttarakhand. This processes 10 tons biomass for making 2G ethanol. So that's I will not be talking much about the 2G ethanol, because there are other speakers who would be discussing about those, but just to give that what the center is doing. So just wanted to give you the idea. Okay. So the platform technologies which we are working with is, one is agricultural waste. Okay, so agricultural waste is something which is going into 2G ethanol. And we are also working in municipal solid waste, municipal liquid waste, carbon dioxide and solar energy, everything. So what is typically what is our mandate, or I would say our ideology is that we have carbon we have carbon which is present in multiple forms. Now this is waste. Agriculture waste is something what we are treating as waste. There is a carbon. Okay. Now, as I told you that why we have to think differently and why we have to uh, you know find our own solutions, and why we cannot just adopt the solutions which are present, or which adopt the solutions which US or UK has identified. The very reason is, let's come to the agricultural waste. Agricultural waste. will go to ethanol. That's something the country is targeting. Now the problem with us is our in case of US they grow essentially corn. Brazil they go grow corn. That's all about it. Okay, but in our case we have feedstock. Our feedstock varies. In South India we will be growing paddy. In North India we will be growing wheat. In mid central India we will be having bagas, that is sugarcane. We will in Gujarat and those sectors we will be growing cotton. Then we have in Madhya Pradesh, we will be having soya because there the soya production is very high. In Assam again paddy. So our feedstock is not the same. It changes. It changes, and also we have crop rotation also within North India also. We you must have heard about New Delhi issue wherein they are getting a lot of pollution because in between the two crops they are taking paddy crop. So we have crop rotation also happening. So the technology which are being made by US, those are typically based on corn. Okay, they do not have any feedstock variation over there. Whereas for us, it has to be feedstock agnostic. So and because I hope that that earlier speaker must have told you that whenever I'm making any biofuel out of such kind of waste, it is going to be a kind of a decentralized plant. So we will have multiple plants across the country and not something localized at one point. It's not because there is no point of carrying the paddy waste all the way to Madhya Pradesh. No, that's not happening because there is lot of energy being will be wasted in carrying that, just carrying the feedstock from point A to point B. That's it has to be in the radius of 5 to 10 kilometers. That is something is affordable. Otherwise, it wouldn't become sustainable. Okay, so we have to. That's the reason the government of India realized it long back, and that's the reason they funded different kind of centers to work on such technologies where we have to build our in-house own technologies, which would so the plant has to be based on whatever crop I am getting. So if it is in Gujarat, it would get paddy for some time and then it would get cotton stalk for some time. So something like that has to be worked out. So that's the challenge we have. So there are challenge, but of course you have the moment you have challenges, you have opportunities also. So that's with the agriculture waste. There is one more aspect to the agricultural waste also, that we use lot of our agricultural waste as the fodder. So that is something which will not be available for us, because if it goes for animal feed, then it will not be available for making 2G ethanol. So that's again one more challenge which we would have. But of course, after going for fodder also, there are, because cotton stalk doesn't go as a fodder. Okay, rice straw doesn't go as a fodder. The very reason is there is high silica content over there, and that's why animal they do not really like that, and that's the reason why that will be made available for 2G ethanol and such kind of applications. So that's the story with 2G ethanol. India is coming up with their 2G ethanol plant, and very soon that will be. We are still importing, even if we have 20% mandate for blending, we are not able to fulfill that mandate. Right now, the mandate is being fulfilled using 1G sugar, that is from molasses. Now they are making to bioethanol, and that is being used for mixing. But very soon we will have agricultural waste also coming in, and we will be using our agricultural waste for making 2G ethanol, and that's how we will try to become self-sufficient with respect to 2G ethanol. But that's the story. That's something which is going to be our immediate requirement. But in addition to that, we also have, what we are doing right now is try to fulfill our immediate need. But we will also have to look at the future, our future needs. Okay, future needs, especially to with the kind of climate change we are facing. So it is like it is written on the world that climate change is here. It is not happening. It's happening in sooner. It is there. We all are facing those issues right now to and there is no way you can arrest it right now because the ball has already got set and has been rolling. It's just that whatever best we can do to minimize its impact. For the moment we have to first think what can be done to minimize the impact, and then the moment we are able to arrive there, then we can see that that can be mitigated. The best way of doing is the municipal solid waste and municipal liquid waste. So that I have seen that in your program you have people who are talking about municipal solid waste and municipal liquid waste. I wouldn't be getting into those details, but I will be talking about solar and carbon dioxide. Municipal solid waste and liquid waste. So the challenges over there is the carbon is in a very dilute form and in very heterogeneous form, and that's the challenge over there. How are we going to trap them? So that's going to be the big challenge. Uh, the idea over here is so whatever carbon dioxide waste, may it be in agricultural form, may it be in municipal form, or may it be in municipal liquid form. The idea is that you have to make it as the utilizable carbon. You should be able to harness that carbon and then take it and make it in such a way that it is now the carbon is utilizable. There is lot of carbon present over there. It's just that how to make it convert it into the utilizable form sustainably and in with the commercial viability. That's the challenge which we are facing. Carbon is there, that's not a problem. It's just that making it utilizable form. The moment you are able to get it, any of your waste, the moment you are able to get it in utilizable form, then you can have very smart chemical biotech conversion technologies, and you can then take that carbon into food, feed, energy, material, chemicals. You can actually divert it in any form, the way you want to use it. But the challenge is it is in a very diluted form. How to harness that and how to make it utilizable. Moment I am able to get it in the utilizable form, sky is the limit. You can actually, practically, you can convert it into the way you want. Uh, so the center has all these different sub departments, I would say, that we have as I told you that you have to work together, and that's the reason why we have developed such kind of in-house expertise wherein we are working in the area of green chemistry catalysis. We have separation technology group, algal biotechnology group, synthetic biology group, which is looking at the future uh, biofuel technologies, enzyme technology group, because that is something is very much required while when you want to make the carbon in the utilizable form. So that's the area where we are extensively working. And fermentation, again, as I told you, sky is the limit. You can take it in food, feed, and different forms then, but that is something which will happen through the fermentation route, and that's the reason we have in-house expertise for all these areas. Okay. So as I told you that how to get it in food, once you have the carbon, you can make proteins, you can make fatty acids, fat, you can make organic acids, alcohols. Alcohol is one of the thing, but you can get it into polymer, then liquid fuel, feed, and specialty products. With a growing population, something which is not being discussed is because we are talking a lot about the fuel and bioenergy and those areas, because the resources are depleting, and we are facing issues of huge import we are doing, and we have to be self-sufficient. Something which we have not looked into as a country, we should also look into how are we going to feed our next generations, because that is something where we will require protein and oil for feeding our people, as well as the animals. That's something, because the two, because if you see, the country is very poor with respect to protein consumption, and to achieve that kind of protein consumption, we should be able to start growing more protein. Unfortunately, with the current agricultural practices, we will not be able to make more proteins with that. Okay, we have reached to, you can always say we have reached to a stagnancy. Okay, wherein is we will not be able to make anything much more than whatever we are making right now. But the population is increasing, and so the requirement, the demand is going to increase. How are we going to feed our people? And similarly, if you are looking at protein, the protein source is again coming from your fish and your chicken and those kind of products. Even you require protein to be fed to your fish and to your chicken and so on. Where are you going to get those proteins? So that is something is very hard pressing right now. That's not the focus area, but that is going to be the, again, the way we are right now, finding it difficult to manage our fuel. Similarly, we will have these issues coming in next decade. So that's going to be the next decade problem, which to which we have to start finding solution right now. So the the mandate which was given to the center was making 2G ethanol, and then there was another mandate which was given to us, that is looking at the advanced fuel, which is going to be the algal based fuel. Now let me tell you first thing, as a kind of a disclaimer, that algal, any fuel coming from algae is not happening right now, and it will not happen even in next decade. Okay, so that is something which is not happening right now. We will have to be dependent on other resources and other technologies to get our fuel. Algal biofuel or fuel coming from algae would happen only after we start cultivating algae for our food and feed application. Okay, the moment, because algae, I will tell you in next few minutes that why am I saying so, that it is why it is not happening right now. Let's have a look at it. I'm skipping some of these slides because we would have. I would like to concentrate more on the upcoming technologies. So if you see that for 1 hectare, the biomass, this is the lignocellulosic biomass, which you can get is maximum 100 tons, which will give you 5 tons of biofuel. Okay, now if you look at and it can also give you, the 1 hectare can also give you 5 tons of vegetable oil, which will give you 5 tons of biodiesel. But if you start using that particular area for growing algae, 1 hectare area for growing algae, it would give you around 25 tons of algal oil, and that can go into, that can give you 25 tons of biodiesel. It sounds very rosy and sounds very promising. That yes, getting 5 times more per hectare, perfect. It's a very, very good situation. But the only thing over here is this has to be commercially viable and sustainable. Our earlier speaker did mention about the sustainability, and I would reiterate that the sustainability is something which cannot be neglected, because this is possible. I'm not saying that this is not possible. This is very much possible, but at what cost? Okay, because that is going to decide the commercial sustainability. The very reason is, if I'm making something, uh, I am making fuel over here. Biodiesel is something which I will have to sell at say 60 rupees or 70 rupees, you know, per liter or something like that. So in that case, if I have to sell it at 50 or 60 rupees per liter, then I should be able to make it at 30 rupees. If I am able to make it at 25, 30 rupees a liter, then only after transportation and all other cost uh, combined, I will be able to sell it at 60, 70 rupees later. Is it possible? No, that is something not possible. Making, yes, I can make 25 tons per hectare, but I will not be able to make it at the commercially viable cost. But I will come back to these slides. Okay, so this is the scenario that you can because algal biofuel, you can, sunlight is there, you can, carbon dioxide is there, you can select any of the algae and you can get biomass, and then the moment you get biomass, you can get algal oil, proteins, carbohydrates, and biomass. The biomass can directly go for firing, or else you can take algal oil and convert it into jet fuel. You can convert it. Proteins can also be converted into biodiesel, and carbohydrates can be further fermented into ethanol and other biofuel. The biomass cost, the productivities, the current productivities are just 60 to 80 tons per year per hectare biomass productivities. How much biomass you can get is around 80 tons per year per hectare, and biomass cost is 100, rather it is, I would say it is more than 100 rupees per kg. And what should be the cost? The biomass cost should be less than 10, and the productivities have to be 100 and 200. If I am able to make 100 or 200 tons of algal biomass in per hectare per year at the cost of 10 rupees per kg, then only I will be able to take it for biofuel. Otherwise, it is not sustainable and not commercially viable. That's the current scenario. Okay, having said so, then why we should be looking at algae? Why we should be looking? Then algae is something which is not going to be viable. Then why we should look at this? Look at this for look at these candidates. Now I'll tell you that why we should be looking at algae as a country. Why we should be looking at algae. Or I am not sure whether anyone of like all of you are aware of this, but I will tell you why we should be. Look at this. We have been blessed. This also we are been blessed. So this yellow is something wherein the light intensities are good, okay, and the sunlight. So you get throughout the year you get nice sunlight because that's the food. Algae are nothing but photosynthetic organisms. Whenever you are traveling, you get those green ponds. So you get those green ponds. So there you can see those green ponds. The green color is because of the algae. These are microscopic organisms. These are photosynthetic organisms. So they require sunlight, they require carbon dioxide, and they would require some nutrients. They are like plant, you can say water plant. But there you have two types. You have uh, yeah. So you have this kind of a microscopic organisms, and you also have such kind of a plant-like structure, which are macroalgae, and then you have microalgae. Okay, so there are different, about 70, more than 75,000 different algal species are present in nature, and in case of seaweeds, also similar kind of biodiversity you find, macroscopic macroalgae you find in nature. Why country? Because as I said, light is available for us. Another resource which we have abundantly present, and that is something you can say freely available. Light is also free for us. We don't have to pay. Sunlight is free for us throughout the year, and a nice sunshine we have throughout the year, and all parts of the country. It's not that just only part of the country. You can find anywhere in the country throughout the year we have light availability. Also, we have such kind of a huge coastline. You will have almost 75, 7,000, sorry, 7,500 kilometer coastline. Okay, so that is something which is available, and the land next to sea is usually the coastal land is non-agricultural land. Okay, so it is non-agricultural land. There is no agricultural activity which happens, intense agricultural. There would be some, no small scale agriculture may happen, but having the high salt concentration, it is not a very fertile land, and that's the reason this land is available, and the seawater is available at no cost. Okay, so that is something the country should be leveraging. So we can use this land, light resources, and this water for cultivation of algae, because that is something we will be able to do in very less time, because we do not have, you know, because there is no competition happening over there. If anything you want to do in any other part of the country, you will always have, competition, because that's the food versus fuel competition, wherein I will not be able to grow anything for fuel or for any other purpose, because all my land is agricultural land, most of the land, and wherein I will have to grow food. But yes, this coastal line or the coastal area can be tapped for growing algae. So that's the advantage as a country we have, and we should use that to our advantage. Why algal feedstocks are advantageous as compared to the terrestrial feedstock? First thing is they have higher growth rates than their terrestrial cousins. Okay, they grow very fast as compared to terrestrial cousins. Okay, they do not compete, as I have told you, they do not compete with their resources with the conventional agriculture, and you can use brackish water. You can also use sewage water. You can use, of course, you can use sea water. It's just that you have to use the marine species, but you can use sea water and you can grow things. Non-arable land can be used. If you have any waste stream coming, waste carbon dioxide, not waste stream, waste carbon dioxide stream coming uh, to this coastal area, that can also be channelized, because algae being photosynthetic, they would require, they would prefer to have carbon dioxide. That's their food. Okay, the biomass is homogeneous and it is without lignin, because processing of biomass for any fuel application, lignocellulosic biomass, you have to deal with lignin, which is very, that's it's a carbon rich entity, but very recalcitrant and very difficult, and it imposes a lot of challenges. Whereas this particular biomass, whatever we are getting using algae, is lignin free biomass. Again, we can do the round year cultivation, because our Indian seas are warm. We do not, the way you have, as you go towards north side, okay, those subcontinents, they face, drop in the temperature, because algae, like any living organism, they would prefer temperature between 20 to 30. Okay, they will grow the best, and our seas are, reasonably well maintained with respect to temperature, so we can use sea water throughout the year for growing algae, and then biomass can be harvested during all season. Okay, so there is, maybe during, even, and also I will show you a few examples where, the biomass can be harvested, or rather you can even continue working in monsoon also. So monsoon, the system which you see over here is a kind of an open system where monsoon cultivation is not possible, but we work with closed systems wherein the monsoon cultivation is possible. Okay, what you need to know. So, and this particular algae we are cultivating not for fuel. Please remember, we are cultivating them for not for fuel, but to feed our to to make food and feed. How that is possible, we'll see in some time. Okay, but to arrive there, what all things we need to know? We need to know very. We should have good understanding for algal cell engineering, algal growth engineering, and algal downstream processing. Uh, these are all at nascent stage, at very early stage. Whenever I'm saying cell engineering, it's nothing to do with genetic engineering, but to understand algal cells. See, we have good experiences of few centuries of cultivating paddy, wheat, and all that. Similar kind of expertise and understanding needs to be developed when it comes to algae, because they are going to be our next generation feedstock. So similar kind of understanding will come. Now that will come only with practice, because we have not uh, all those practices um, for our paddy wheat has come over the years, or rather the expertise has come over the years, and similar kind of requirement is there for algal cultivation. Couldn't come overnight, but yes, as and more you practice, more and more you grow, you will get to know the challenges, and you will also have the solutions. Okay. So, technological challenges, biology is not very well understood. Large scale cultivation is still not there. That is something in lab people have been growing them for years, but large scale cultivation, field cultivation is still under very early stage. And once the biomass is there, how am I going to process it and converting? So conversion technologies, whether I want to use it for food, feed, or any other application for biomaterial, how is that going to be achieved? So those kind of downstream processing, the chemistries are still to be worked out. So this is some things, but the challenge is going to be making biomass, even for food application, even for feed application, making biomass at rupees 10 per kg. Okay. Now we will see that why there is so much cost going in whenever it comes to growing algae. Look at this scenario. Algal cultivation, the cost is 70 to 100 rupees. This will be followed by harvesting. There is substantial cost in harvesting. Then drying the biomass, and then if you are looking at biodiesel, then extracting the biomass. And that's the reason the algal oil is coming at very high price. Now you can see the cost contributors are coming from algae cultivation. So you have to make the cultivation, sustainable and commercially viable. Large scale cultivation. So as we said that there is a huge cost going into cultivation. The large scale cultivation is, as I told you, the sunlight is present, but the sunlight has to be fed very nicely to the algae. Each and every cell should be able to get good exposure to light. So light is there, but the 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 exposure of every algae to the light is something very important, because otherwise in the big, if you are growing something in a big reactor, please imagine you are growing algae. They are kind of a suspended material, and you are growing algae, and they are into a big reactor, which is now the reactor has to be transparent. Okay, if it is not transparent, then it should have huge surface area. The very reason is the light has to be, each and every cell has to get exposed to light. That is something, because it is a photosynthetic organism. Light exposure is very much important. Now if you do not have good light exposure, your productivities will go down, and you will not be able to get good number. Production would go drastically down. Okay. Now, most of the times, whenever people are growing algae, they say that you can use as flue gases, because that's the cheapest source of carbon dioxide. Now, please remember that flue gases would be available near the power plant. Okay. Now, it is not necessary that you will have, because for this kind of scale up cultivation, you require land. Okay, large scale cultivation, you require hectares and hectares of land. Now, not necessary that next to the power plant you will have such kind of land available, not necessary. So also using flue gas for carbon dioxide supply is something may not work every time. It may happen at few instances where the land is available and you can have flue, you can have algae cultivation next to the power plant, but not necessary every time. So you need to have another way of, capturing carbon dioxide and feeding it to algae. The more carbon dioxide you will feed, and they have huge capacity to capture also. That will also mitigate your the climate change effect also. But for that, you have to make some engineering solutions so that the carbon dioxide can be concentrated and can be supplied to algae so that they grow at much higher rate, higher water footprint. Okay, so the footprint as of now for algae cultivation is very high. So there has to be some strategies wherein you are able to either use um, uh, the fresh water, because fresh water cannot be used unless you are recycling it 90%. That is something is the challenge, because you should be able to recycle. If you are using fresh water, then you have to recycle 90%. If you are using marine water also, because you will be taking it from sea, so there also you should be able to recycle your seawater. See, because whenever I'm taking anything from sea, there is a pumping cost. It is the water is available at no cost, but there is a pumping cost involved over here. So I have to pump in that water for couple of kilometers wherever my farm is. Now there is a cost involved over there. So I should be able to recycle that. So that's something one has to realize, that that is something we should be able to manage. Also, there is a high demand for fertilizer, because algae, as you have to give good fertilizers to your plants, similar way, you should be able to give good fertilizers to your algae also. So that's something is the operating cost one has to look into. So recycling strategies are not well established, which needs to be well established if you want to make this as a commercially viable proposal. And also, as I told you that people have been working on few algal species, but these algal species, they work very good in their laboratory, but they actually do not perform when it comes to the outdoor cultivation. Those have been our experiences where we can see that things are absolutely fine when you are working in the laboratory, but not when you are working in the field, real time testing. Okay. So for scale up cultivation, what should be managed? We should be able to manage light, fertilizer, and carbon dioxide, so that only we will able to get the cost effective cultivation of algae. Now please remember, this is something not independent, because they are actually influencing each other. If light is well managed, then my fertilizer requirement will increase, because then if light is well managed and if I am able to supply good carbon dioxide, the algae will start growing faster. They will have higher demand of fertilizer. Similarly, as they are growing faster, they will have higher demand of carbon dioxide. So these are interdependent. So whenever I'm scaling this up, this has to be managed very well so that I will be able to get cost effective cultivation. Okay. This is something which I have discussed. So I will skip these slides. Now the cultivation challenges. I'll tell you the when the people started working with algae, the trend was using recipe pond. Sorry, the trend was using recipe pond. Still, people have been practicing recipe pond for different value addition compounds, because algae are known. So you must have heard about spirulina, which is considered to be superfood. And this spirulina is very rich in protein. It contains almost 60% protein. And we should feel proud that India is one of the biggest um, shareholder, or it has biggest share in supplying spirulina to world. EID Parry, and there are such kind of ventures. EID Parry coming from South India, so they're coming from Tamil Nadu. So they have huge farms in Tamil Nadu, and they are making almost few thousand tons of spirulina in their farm, and they are capturing almost 50% share, global share I'm talking about, of supplying spirulina across the world. And their spirulina is basically EU certified, so that they grow them organically. Okay, so these are the cultivation systems which are typically used for growing algae. You can see that these are open ponds now. If I'm sure you can see these are small ponds. So here you start building your inoculum. From here you go to this scale from here, because that's kind of a progressive thing. You cannot start directly from this reservoir pond. You start from very low quantities. So from laboratory it will come to this, it will grow, then from here it will grow here. So from here and from here it will grow to such big ponds. Okay, but these are open ponds, whereas these are closed. You can see these are closed. There is a kind of a plastic kind of a covering over there. So some sheet is being, it's a kind of a greenhouse kind of a thing. But this is something which is allowing light to pass in. That is something very important, because if there would have been some kind of a typical mesh which is typically used in greenhouse gases, this wouldn't get light. So these are closed. These are again some advanced, I would say, raceway ponds, wherein you can see. So these raceway ponds is the criteria is that you can see paddle wheel over here. You can see paddle wheel over here. So this is something which will keep moving the water, and your water will move in this particular direction. Now look at this. Just these are very much preferred because they are low in capital cost and low in operating cost also, because there's hardly energy is required, because you just have to rotate this wheel and this will take care of the entire circulation. This is something, is a constructed pond. So these are kind of a cement kind of a raceway pond. But this is something where they have put lining. Okay, so they have made dug uh, some pit over here, and then they have put some kind of lining, and then they are using, this is a paddle which will make the necessary movement is something very very important, because the movement decides the exposure to light and mixing of micronutrients for it to grow, and also carbon dioxide supply. So that is something is required. But this particular movement is not, or this particular agitation is, I would say, very poor, because what happens in this, that there is a good movement over here, but then as you go along the length, you will find that there are some dead pockets where your algae may settle down. And if they are settling down, then in that case, if they are not exposed to light, then they will go into, from photosynthetic mode to, they will go into the respiration mode, where they will start consuming whatever carbon they have fixed. Okay, and that will reduce my productivity. So this is something. So there are drawbacks, though these are capex and opex intensive. I think I have the comparison, that raceway pond, and then there are, I will show you the reactors first. So these are the raceway ponds. These are the flat panel bioreactors. So world is moving to these kind of reactor systems. Why, I will tell you why these kind of reactor systems are required. So these are flat panel reactors, and these are the tubular kind of reactors which are typically used for growing algae. Why this kind of closed reactors are required? First is the light efficiency is good, because the light exposure is good. So then you will have, gas transfer is good, reasonably good, and the temperature control is also very good, which is very. In reservoir pond, you do not have such kind of controls. You cannot really control things in case of reservoir pond. They would be, open to sky, and the mixing is poor, so the light exposure is poor. Temperature cannot be maintained. Gas transfer is poor. The and that's the reason you will have low biomass concentration. Biomass will not grow very good, and also you have issues of crop crash, or you have issues of contamination issues because it is open. Control, there is no control, and it is open, so you have issues of contamination issues. That, but it is still preferred because it is low in capital investment and operating. But if we are looking at growing this particular algae for food, feed, and other value added compound application, then tubular or flat panel reactors are something which would be preferred. People are working on both the models, flat panel as well as such kind of tubular reactors, because each it, they have their own pros and cons, but yes, I would say that people are still toying with which one would work better. So even at our lab we have worked with both the models, but then we continued with tubular photobioreactors because we found that was performing better. But yes, the cost over here is high, but volumetric productivity is very, very high. The biomass, which you can produce 3 or 4 times higher biomass per liter per meter square when it comes to these kind of reactor systems as compared to reservoir ponds. Okay. Energy demand is also not very high. It is comparatively low when it comes to the reactor systems. These are the rest ponds and these are the reactors. This is something latest which I could get from the internet. So these are the vertical. If you can see, these were the horizontal reactors. These are the vertical reactors. These are all tubular, but these are horizontal. But here you have your land footprint is going to be very, very high. So this is better because here you have the lesser land footprint. Now, if you see, this is open, but if you see, this is again in the shade. You can see there is a kind of a closed environment over here, and then you have inside that lab you have these kind of reactor system. Now, the reason this, because in India we do not have such kind of reactor system being installed. So all these pictures are from Europe and US, because we don't have such kind of system being installed over here. A few labs, yes, but not at such kind of higher scale. Terry is working very intensively in this particular area, and they are deploying such kind of reactor systems in their laboratory. The reason why you can see such kind of a closed environment, you can see this is better picture. You can this kind of a closed environment over there because they have to deal with drop in temperature, because these countries they have, and sometimes even very low light intensities, because if you go as towards the pole, all these countries they face the low drop in temperature as well as drop in light intensities. We do not face such kind of issues, but yes, those countries to maintain the temperature and to give the additional light, so that's the reason why they have such kind of indoor facilities. But in India, we may not require. We do require some kind of temperature controls sometimes, maybe during summer season, but not otherwise. What happens when it comes to lab to the open environment? Because the translation is something which does not happen so smoothly. Lab, because lab you will be growing algae, under very controlled light and very controlled things, but that's something, the things are very, very harsh when it comes to the natural light environment. I will just take you to the glimpse of Ascati. This is the center which I visited 4 years before. So what happens is this is US one of the largest, I would say, the translation facility wherein they have, so you can see, so on this side of the street they have laboratory, and this is their open translation facility where they have, almost 2 acres of land where they have, test bed facility. This is typically a test bed facility where they take all whatever they are doing in the laboratory, they are taking over there. And in US they have, so the testing will start. So if they are working on a particular strain, that they have 5 such centers across the country at 5 different locations. So they will start their experiment at the same time, on same day at same time, with the same strain. Okay, and those geographically they are uh, placed very far from each other, and of course, as they are very far from each other, they would have their different season and different temperature and so on. So they actually see that how robust is the algal species which they are cultivating, and also to see seasonal impact, geographical variation, what kind of numbers they are getting at different locations. So that's how, and then they will make average of it. That's take care. So that's take care of the sustainability, because if something which works with this particular at this particular location may not work in something at the south, extreme south or extreme north of the country. So that's something they are trying to come up with, and then find out that which is the algal species which needs to be cultivated for whatever application you are looking at. But that's the program they are running under their DOE center. So this is the cultivation facility, which these are the restway ponds. They have everything, restway ponds. Then they have such kind of flat panel reactors. Look at the array of flat panel reactors for cultivating. So this is each is 1,000. I think each is 2,000 liters. So the earlier was flat panel was 50 liter, and then this each is 1,000 to 2,000 liters. These are flat panel reactors which we have. In the bag itself, in the flat panel, they had that HDP bags being placed. Okay, these are the airlift bioreactors. These are polycarbonate bioreactors, which are also again present at Ascati. Okay, what we do at DBT ICT Center. So this is what I told you about, that what people are doing across the globe. Now I will give you the glimpse of what we are working. So I'll tell you, we are working on both macroalgae as well as microalgae. Macroalgae I'll talk little later. First I will talk about the macroalgae, microalgae. So our area of focus. So this is my lab. Again, this is glass house, and here we maintain temperature of the lab temperature is typically maintained around 28 to 30 degrees Celsius, because that's something the algal species will encounter when they will go to the field. And this is open to sunlight. It's just that we have to maintain the temperature. And these are the different reactors, so at different scales. So these are polythene bag reactors, and we so all our algal species are being grown. So we do not work with artificial light, we do not work with low light, but we work with natural light. That's the philosophy we have, wherein, no, because that's the light they will encounter when they will go to the field. So that's the facility which we have. I think this is India's state of the art facility. There are not many places where you will find such kind of facility. So natural light, and you can work at higher temperatures. Work with cyanobacteria, microalgae, macroalgae. We work with all of them. So this is how the lab looks like. It's not a very big lab, maybe 1,000 square feet lab. But yes, so whatever, so we can do actually seasonal variation, because there is a lot of variation happening. So in summer, you will find the light intensities are around 80,000 to 1,20,000 lux, whereas during winter the light intensities would be, they will be around 40,000 to 60,000, 70,000 lux. So you have this kind of seasonal variation. in of course this seasonal variation will be different in different parts of the country, but we encounter in Mumbai, we encounter such kind of seasonal variation. So we work with, summer, we have different algae to be grown during summer, different algae to be grown in winter, and in monsoon we will have less than 10,000, sometimes even 5,000 lux kind of light intensities. And we have our algal species which will be growing in monsoon. So we have studied all these, and we have now such kind of crop rotation happening in our lab itself. Of course, as I told you, we are not working in the area of fuel, but we are using, so we are studying all these algal species for food and feed application, and also value added compounds. So we are looking at, different nutraceuticals, different pharmaceuticals, and, different antioxidant. And we are also exploring these algae for protein, because that's going to be, as I told you, that's going to be our next decade challenge. So we are looking at how can we make algae with higher protein content. That's and also for feed. So something which can go into feed. So that's something we are working. But again, whatever we want to do, we will encounter such kind of outdoor challenges, and that's the reason we are preparing. And each and every species, before it. So there is adjacent over there. There we have our outdoor reactor systems over here, this place, which are open to sky. So whatever we want to do, so every species will be worked in this particular environment for almost more than 1 year, and then it will be transferred outside. The moment we have complete understanding of that particular species, it will be transferred outside for outdoor cultivation. But this is very, I would say, close to the reality, because light and everything doesn't change. As they go out, light doesn't change, temperature also doesn't change. So they do not have any translation shock. The translation happens very smoothly from laboratory to field. Working at different approaches, state of the art facility again, then we look at the robust algal species, and then we actually they'll go a lot of adaptive evolution to thrive into adverse condition. That's what we have realized, and they go a lot of evolution. Whenever we start with any algal species, we realize that after 2 years it behaves very differently. So those kind of adaptive evolution and those kind of changes get accumulated, and and uh, and they work in your for your benefit. Then we also have the different tools by which we can look at the photosynthetic performance, that how much active it is photosynthetically. So that's the kind of a tool which we can use no, to check its photosynthetic performance, because the moment you have drop in your photosynthetic performance, we understand that this crop is it's time for it to harvest, and this crop will not do any better now. So either you harvest it, or take the different course of action which is required. Okay, then lot of work going on into organic cultivation, because if you are looking at food and feed, then organic cultivation is something required. So, and using waste streams, that is something is also being targeted at the laboratory. Mixotrophic cultivation, using different waste streams. This mixotrophic cultivation is waste streams are basically dairy waste or food waste, which can be then converted in, which can be diverted into, for growing algae, because mild amount of sugars are very helpful and they are good for algae to grow. So that's the work which we do. And also bioprospecting for very value add. So right now we are working with lutein, zeaxanthin, astaxanthin, then vegan EPA, and those kind of aspects we are looking at. And water management, that is something I will show you what we are doing in this area. And also the genetic engineering of algae. So basically for improving their stress and those. Now since we are working with closed photobioreactors, genetically engineered algae can be taken to such kind of closed photobioreactors. You cannot grow genetically engineered algae for uh, in open raised ponds. Okay. So again, we have little different conventional approach, because people have been using. So how different we are looking at? Like people have been using artificial light, we use natural light. Oleaginous species, what we realize that even if I'm able to get a good biomass producer, because I need to have something which has 30% oil, or rather I would say 60% oil, but grows very slowly, and something which grows very fast has 20% oil but has nice growth rate, that may help me. So that's the reason we always have biomass producers and not the oleaginous species, because oleaginous species, they that's our experience that they grow little slow. Then oil content without loss of biomass. People have been working on two-stage cultivation. We believe in one-stage cultivation. Of course, there are some strategies by which oil can be improved. And then photosynthetic efficiencies and high-value compounds are something which we look more than oil content, irrespective of whatever application you are looking at. So that's our approach, and we think a little differently than what people have been doing. These are the recipe pond facility which we had. We discontinued all these facility, and then we came to the reactor designs, wherein we tried to got into different reactors, which these are in-house reactors. So we started with such kind of small reactors, and then we came up with different something which you have to do. You have to first try with 10, 15, 20 liters reactors. So these, and then we moved on to, as I told you, we tried with flat and we tried with tubular reactors. Now please remember that we do not have any expertise and any vendor who would do this. Okay, because there is no way we can buy anything from the service providers which are outside the country. And we always believe that we have to find our own solution, and that's the reason we decided that we will design our own reactors. We tried different models. So these are the flat panel, which we continued because of the poor mass transfer, and there were a lot of issues with dead zones. And we then continued with such kind of airlift reactor. So this is 500 liter reactor. Why I'm showing you all these pictures, because there is a lot of, I would say, there is one has to realize that you have to find your own solution, and the solution doesn't come overnight. There's a lot of trial and errors one has to put in, and that also requires money as well as time. And I would say that you would require to have that kind of passion. Only then that would happen, because this is what I'm showing you is almost two masters and two PhD students work has gone in. I'm just giving you the glimpse, but to 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 conceptualize it and then execute it, especially when you do not have any vendors providing such kind of services, to make the one vendor to supply you and then make and something out of it which will work was really challenging. But yes, we worked on it, and then we worked on different. So this is the serial airlet reactor is the model which we identified, which is operation friendly as well as less on the operating cost. It has to be operation friendly also, because you are going to deploy such kind of closed reactor system in remote areas near the coast where you may not have very trained manpower. So things have to be modular. So deployment has to be very smooth. So you should make something which is deployable. If you make anything very high tech, it may not be possible for you to deploy at that kind of a scale. So things have to be modular, and they should be operation friendly, because these are going to be handled by farmers. So they should be able to operate them. So that was the challenge we had, and we made. So this is a kind of a reactor which is now uh, being established at the center. This is 1,000 liter reactor. We have two such 1,000 liter reactor, and as I told you that India has to look for its own solution. So now, if you are able to grow microalgae, then why not grow macroalgae also? So we are using the same reactor system for growing macroalgae. I'll show you how we are growing macroalgae also in this. So that's but yes, we are using such. So these are and here the mixing is not through any pump or nothing. We just purge air. So there is a compressor and we purge air, and then the air is taking care of all mixing and everything. So these are polycarbonate tubes, and these are the HDP top and bottom, and you, of course you need to have some kind of a support system to hold this because this is 1000 liter. Now this is modular, so what we have planned that we will deploy such kind of modular system and use this kind of a reactor system for different and many applications. Wouldn't go into detail, but yes, the biomass productivity is now with this because see the illumination area, uh, the illuminated area to volume ratio is very high, unlike reservoir pond, because in reservoir pond the light penetrates only for 2 or 3, or rather when the culture is dilute, say 3-4 mm, but as the culture gets dense only 1 mm or something like that. Here the dilution, here the illuminated area is good, so you will get higher productivities. So it improves productivities, it provides temperature, nutrient, pH control at desired levels. Not necessary that you have to control based, but if you are working on something very high value compounds where you can afford to temperature, pH, and all that, then if that cost is can be accommodated, then you can always do that because if you control things, you are always going to get good produce. Okay. Okay, now we have worked with these systems. We have also encountered that the seasonal variation impact, because if you see in literature that there is lot of variation in biomass composition and when it comes to the different seasons. So seasonal variation impact is reduced, I would say. It is not completely gone. It is reduced substantially. The cost is less than 100 rupees per liter is the fabrication and installation, minimal operating energy, lower land footprint, modular, expandable, deployable, so you can do it in mega scale, and everything has been established now. This how to fabricate, execute, everything is being established. We'll just take you since the area was biofuel. So after you cultivate, what so you have to provide all these inputs to your algae, and after you cultivate, what you do with this? Once you have this kind of a cultivation system, then what can be done with the biomass? First thing you have to harvest. There are different technologies by which the algal biomass can be harvested. But the best one is either you can go with membrane based technology or flocculation based technologies. That is something the other technologies are not commercially viable, but these two technologies seems to be commercially viable. There are two. Once you harvest the biomass, concentrate the biomass, then you can go for, there are different uh, processes by which you can get fuel. But for biochemical conversion, no gasification and those kind of pyrolysis and those kind of conversion, drying is required, which is and because algae holds 90% water, so that is something out of question because then you have to give energy to get rid of that one, get rid of that water, and that is not going to be sustainable, and that's the reason drying is ruled out. You just dewater, concentrate, process the wet biomass through liquefaction, and you can get bio oil, or you can go for fermentation. That is also we have tried, and get the biogas. So these are the only biodiesel you cannot, because again drying is required, and that is very killing. So that's the reason that route is not being worked, and it is not never ever going to be sustainable. So though there are many routes, you have to choose the right one which will make things viable and sustainable. So bio-oil and biogas are the two routes which are possible, and we have worked on these. Right now we are not working, but we have worked on these routes. This is how the bio-oil. So you cultivate, we cultivated it, we concentrated it, then thermoliquefaction. Thermoliquefaction is nothing but cooking at under high temperature and pressure and giving such kind of a reaction which actually happens inside the earth where your bio crude is being made, because that's under pressure and under high temperature. Similar things you try to simulate and you get bio crude. This bio crude can be then used for biofuel purpose. What is important over here is biocrude. The moment you get biocrude, what you can do is the wet biomass, after concentrating, that's something. Biorefinery concept is something which is very important, because the wet biomass you can actually process for proteins, carotenoids. You can extract these proteins, carotenoids, and then the rest of the biomass you can send for liquefaction for biocrude. So that's the refinery kind of a thing, where you try to capture, couple of other products in addition to your fuel. Okay, so that you can have good, uh, I would say, cost economics, because your proteins, your carotenoids will be sold at much higher cost, and then your oil you can afford to sell at lower cost. So that's the idea over here. And of course, after you do or your extraction, whatever nutrients are there, the water has to recycle, and your nutrients are also supposed to be recycled. Then only it becomes the viable thing. Recycle has to be more than 50% and water more than 90%. The milestones of algal industry have been, see, as you see, for fine chemicals, so this is a kind of a ladder: fine chemicals, human nutrition, aquaculture, feed. But as you are going below the, down the ladder, the price also goes decreasing. Fine chemicals, carotenoids, now some of the carotenoids are being sold at say 50,000, based on of course purity, but 15,000 per kg. PUFAs are sold at 10,000 per kg. But when it comes to oil, it has to be sold at 60 rupees per liter or something like that. So that is something which we have to keep in mind when it comes to, growing algae. So you need to know which application you are looking at, and based on that, you have to work on the different aspects. Okay. So these are the, so as I told you that we worked with in the fuel area, and then we diverted into the value addition area, wherein we are looking at feed, nutrition, pigment. So we have all these algal species with us, and we work on their different potentials and valorization, because we know that why this is important, because for each and everything, domestication of algal species is something which is very, very important. So first you have to domesticate. You just cannot start growing in the natural. So first you have to domesticate, and that is going to be again, uh, uh, it is going to be, country specific, area specific. I would say not even country, area specific. If I want to do anything in south part of the India, then I have to domesticate it to that cultivation condition, and we have actually done that. So wherein we have domesticated it for a different uh, cultivation condition. Then while we are doing this domestication, and while we are doing, trying to explore these algae for different such application, because these are very good feedstocks, okay, and they are going to be there in next few years. While we are domesticating them, we also need to have the cultivation skills and skills. Both are required. That is something, and unless and until we do it, we will not be able to get these kind of things. So take the antioxidant, which are very, very high in price, something which sells at 50,000 per kg. Take that and try to cultivate it at a scale. You will get the hands-on experience how to cultivate it. And then after that, the moment you have skills and scales, skills in handling such kind of skills, then only you will be able to grow anything at lower cost, because with time things will progress, and the cost of production will go down. It would happen as the scale will increase, the cost of production will go down, and then the biofuel can come in reach. Then only we will be able to make anything for biofuels. Not at the moment, because we do not have anything over here. We do not have anything over here. The moment we have all these in place, then the biomass, and we need to also know how the biomass can be valorized. I can get the multiple products out of it, and then rest of the things I can push for biofuel. So that is something, if I'm able to work in next decade on all these aspects, then only maybe after 20 years we can say that we can have biofuel coming from algal feedstock. Okay. This is what we are doing in the area of algae. I will take you one more quickly through, uh, how much time do I have?
Madam, maybe 10 minutes,
Ma'am,
10 more minutes would do, madam. 10 more
Okay,
minutes.
so yeah, 10 more minutes because I know that I'm in, I am between their lunch. So just give me, I'll take 5 minutes on this and 5 minutes on macroalgae. I'll just take you there.
Sure, madam.
10 minutes, yeah. So demonstration, so what we are doing at ICT that we are looking, as I told you that you have municipal liquid waste where the carbon is in dilute form. And algae are very good candidate to treat sewage water. So that's something which we have worked and now we have working on municipal liquid waste wherein what carbon dioxide you have sewage water which is just a minute. Sewage water is, it has NP, but it is not very rich in NP. We always say that sewage water is rich in NP. It's not very rich. It is at very low quantities because it is in very dilute form. But what we thought that if we have the reactor system in place and if we have all these things, then we should be able to clean water and we should be able to, of course, carbon dioxide will be converted into clean air and will not get energy fuel, but energy fuel was something we targeted, but right now I don't see energy or fuel coming out of it. But at least if we are able to clean the water and that is fresh water, that's also good enough incentive for establishing such kind of technology. Whereas if you see the other treatments consume oxygen, but whereas and produce carbon dioxide, but whereas we consume carbon dioxide. I would not get into details, but this is the schematic which we have. So you have effluent and you have the reactor system and you will get it clean. This is something which we have worked at 1000 liter, and this is how our technology works as compared to the other prevalent technologies. So we have worked with worked with few engineers to establish this particular technology, but we are much better than as compared to other technologies. And the beauty of this particular, so this is what we did. So we have costed. So we collected this is a 10,000 liter collection tank of municipal liquid waste. We collected that and then we pushed that particular to our reactor systems and we tried to see that how the water can be treated. And we actually saw that water can be treated using this reactor system and municipal liquid water can be treated. You can see this is the comparison, and the best part was, and everything was in permissible limit. And the best part was that it even didn't have any coliform. So the model which we are now establishing is for housing societies and housing complex. This is a kind of a decentralized model where on the top of your terrace housing society this model can go, or in housing complex you can have a piece of land where this can be worked. And then we have also worked on that how much, because each society would produce so much amount of water, municipal liquid waste, how much reactor system or what capacity reactor system is required. The best thing which usually see algae has been known to treat municipal liquid waste, but you can see the HRTs or the residence time is usually they are in days. So 6 days, 7 days is the treatment time. What we have is around less than 10 days. 10 sorry less than 10 hours, and the very reason is we are working with photobioreactor system where the light and everything is well managed, and that's the reason why we are able to reduce the HRT. So that's something which we are taking these are the different, how much would be the cost per person, because something which in Mumbai we have to pay if I'm taking any parking parking space, then I have to pay for that, and that runs into few lakhs. So the similar model which we are looking, that per person you will have to give, while we are purchasing any flat, you will have to give so much of so some 10,000 or 20,000 cost. That's the fixed cost, or that's the one-time cost, and then in that we will be able to cover the capital investment for the reactor building, and rest of the thing can be sustainable. And the water can be used for all sanitary purpose. So you will be able to recycle almost 90% of your water, because the sanitary, and you will be able to use it for sanitary purpose. We have worked with, if you just put one RO system, then you can even take for drinking purposes, but of course we will not be getting over there. It's just that sanitary purpose. Even if you are able to achieve the sanitary purpose, that's good. So we have advantages. We recycle, we remediate, we recover NNP, which was present in municipal liquid waste, and we reduce the carbon dioxide, because algae, we don't have to provide any carbon dioxide, but air will be purged, and whatever carbon dioxide is there in the air will be captured by the algae which are growing in that reactor system. So this is the reactor system. So there is one plant which is coming up to treat 2 lakh liter per day. So this is a, you can see the 50,000. So this is how the algal farm is. This is under installation. This is under installation, and we will be soon commissioning this plant. This is at the residential complex of BPCL, and we will be using their complex municipal liquid waste. So that's what we have been working on, municipal liquid waste. And there is one more thing which I would want, that what we are doing in the area of macroalgae, because macroalgae is something, if you go across the globe, that's something making a lot of noise, and people are talking a lot for the climate change and for next generation, feeding people, and taking care of greenhouse gases. Macroalgae is something which is going to catch up. Again, if we do not start working in the area of macroalgae, we will miss the buzz, because macroalgae are typically sea-based, and we have the coastline. As I told you, we have the coastline, we have the light, so we should be looking at macroalgae. Unfortunately, in India, so we have green, brown, red seaweeds, which are commonly typically being used, and they have been there. We do not eat them because that's not in our staple diet, but Asian countries, you will find the kind of skin all those Chinese and Japanese and Korean girls have. That's because of the kind of food they are eating, and they eat all these seaweeds because these are superfood. So seaweed, they are there in the food, not in India, but Europe, US, and Asian countries. It's already there as a food. And in Asian countries, it's a staple diet for them. Uh, why seaweed is the next food? Because it is rich in protein, vitamin A. You name it, and it's there. You just name it, and it's there, and that's the reason it is going to be the superfood. Of course, India is not there on the map in case of cultivation. I'll tell you, in India, the macroalgae cultivation happens in Mandapam, near that's the south coast, and near Gujarat. The reason is, why not happen in Maharashtra? Why not in Kerala? The very reason is our sea is very, very rough. All the Asian countries which are eating this kind of superfood, they have very calm sea, and we do not have such kind of calm sea. Our slopes of the seabeds are very steep. In case of Asian countries, you can walk on seabed with at a knee length sea water for miles and miles. We hardly can do that. And that's the reason why we do not have such kind of um, seaweed farming happening in India. Then what is? But we have resources, because we have everything. So what can be done in that direction? I just tell you that where we encounter this. So food, as I told you, that we do not eat that in our food, but but we eat all this. So carrageenan, which is coming from the red, and believe me, in South India, you have many cottage industries which are making. So we import the seaweeds, we import this Kappa ficus, and because that's something which is being made in Indonesia and those countries, Asian countries, the biomass is imported. We make carrageenan. India makes process that biomass. We make carrageenan, and then we supply to the world. That's how the model is working. So why not produce such kind of kappa ficus in our country? Carrageenan, these are the applications, and you name it, any food preparation, feed preparation, you will find carrageenan is present. Then alginate, which is coming from brown seaweeds, gelling, binding, stabilizing agent, and then lot of organic farming is now coming up with this ascophyllum fertilizer. Then it's there in many applications. Wouldn't get into that because I'm running out of time. And then there is one more phycocolloid coming, which is agar agar. We all have been using it for different applications. And in addition to that, there is huge market coming up for fertilizer, soil conditioner, cosmetics. It's already there in outside India. It will catch up very soon in India as well. So these are the applications, and then what can be done. And again, they can work on the biorefinery molecule because single biomass can give you, multiple products like this. So what needs to be done? As I told you, we have very good coastline, and we have very seaweed rich beds. So that is something we should be, targeting. This is something, again, it's going to be eco-friendly activity, and the seaweeds can provide uniform quality for the industrial. Now, what is typically being done across the globe is, such kind of sea-based cultivation. So you actually grow in them in sea. These are raft, these are mechanized, these are long line. These are kind of a sea-based cultivation. This is not from India, but this is from Japan and Indonesia. So this is how you can actually see the seabed and you can walk. So this is something the biomass. But the challenges in sea-based cultivation is abiotic stress with the climate change, rough sea. So that is something is a very, and then there's loss of crop, limited growth season because India being the monsoon faced country, we will have limited season. Grazing, seaweed diseases is something which is coming up, and then it would definitely have. And we have biodiversity board, which is stopping us from getting into such kind of seabed cultivation, because then that is affecting the local flora and fauna. And we should also learn from our mistakes, because that's what happened in our terrestrial. Our hybrid versions are now taken over by all our traditional crop varieties. So whether it is good, bad, that's something debatable, but that is something. But those are the lessons which we have to take, and that's the reason biodiversity is board is not letting this kind of activities coming out, sea-based cultivation um, coming out as aggressively as it should have been. Uh, so again, coming to the land-based cultivation, there is one company, Secura, you can check on the internet, the Secura, which is doing this kind of a land-based cultivation. They are doing it in tank, because there you can control things. In sea, you cannot control things. You cannot even provide fertilizer in sea because it gets diluted. But in case I'm doing it on land, where I'm using the same resources, same seawater, but just on the land, then I can get a consistent and high biomass yield, because things will not be diluted. I can get completely hold on the biomass which I am getting. If I'm wanting more in phycocolloid, I can produce such kind of biomass. Control over cultivation system, relatively unaffected by seasonal variation. Otherwise, the biomass is very affected by seasonal variation, and then you can protect them from aquatic cases. What we have done is just 2 minutes. What we have done with Ulva lactuca, that's the green seaweed which we have worked. Again, very rich. So these are the nutritional potential of Ulva lactuca. We actually domesticated it. We got it. We domesticated it. This is almost, I'm telling you, the which happened over the period of 5 years. Nothing happens overnight. And this is how we did it in the lab. And then lot of work going on. And then we also established the biorefinery, wherein we got different products out of it. And I will show you. This is a Secura which is also working on the same. I will just want to show you the. So this is how we are growing them. So this is how the reactor works. So we are purging air, and this is how the ulva is growing. This is no one is working in this particular growing macroalgae photobioreactor. This is a full view and this is a closed view. And everything is mixed using air. So this is, and we are able to get, very good rich productivities. And this is like this, we have published a paper, so you can just Google and see. Very recently it has been published, and you can see that the seasonal variation and everything is being taken care of this. So this is the online cultivation which is happening. This we did on terrace, it was very challenging, but then this can be deployed, next to the sea coast. This is a refinery which we consider, and there is another aspect of genetic engineering of algae. Wouldn't get into this because there's a short of, I'm already crossed my time limit. So that's something is the next generation. But to arrive here, to reaching to this, whether you are going through the metabolic engineering, genetic engineering, tools needs to be developed. You can't. People have been working, but even if you have any genetically engineered which is performing very good, what you would require is this. These are going to be challenges because the scale of production which you will be able to achieve and the environmental sustainability and economic viability, that is something will be hard plus. India has capacities and infrastructure. India has been investing and it should arrive for algal cultivation. So I will thank DBT, DST and DSR for funding and my teams of masters, PhD and postdoc students for giving me such kind of material to discuss and share with you. Thank you.
Thank you very much, madam. I am Ruben Sudhakar. On the hindsight, I think we as event organizers must have allocated you two consecutive sessions so that you could have fully covered the work. You have so much to say about algae. We would certainly plan for one such a consecutive session in the next workshop. We would love to hear you all these that you have got in the slides. I hope all the participants have got enriched on algae, and I think the kind of value-added products, the cost that you told, has really started making us think about making them in whatever little quantities we could make. I think many of our participants would also be interested in doing it. Thank you very much once again for that wonderful and informative lecture, madam. Thank you very much.
Thank you. Thank you. My pleasure. And if anyone has any queries, they can write to me anytime.
Thank you.